inside an unfertilized mammalian egg, a full 10% of the entire cell's volume is it's taken up by this massive, totally mysterious structure that we've honestly never been able to clearly see before.
Right. It really was a mystery. For decades, it just showed up as these blurry, fuzzy lines under electron microscopes.
Yeah, just these vague streaks. But we knew one terrifying fact about those fuzzy lines. If you knock out the proteins that make them up, the egg can literally never develop into an embryo. It just stops.
Exactly. It's game over for the cell.
So today, we are opening the ultimate microscopic survival kit. We're taking you on a journey inside the absolute beginning of mammalian life to figure out, exactly how a mother packs the essentials for a new embryo.
It is genuinely the biological equivalent of finding a hidden, highly fortified city inside a single cell. I mean, the fertilized egg, the zygote, it is completely cut off from the outside world at first.
Right. It has no external supply line.
Exactly. It relies entirely on maternal stores that are prepackaged in the oocyte, which is the unfertilized egg. And if those stores aren't packed with absolute atomic precision, life simply doesn't begin.
Okay, so let's unpack this. Because to understand that precision, we are doing a deep dive into a really groundbreaking 2026 paper from the journal Nature. It's titled Structure of the Mouse Cytoplasmic Lattice.
And what these researchers accomplished structurally is, it's almost unthinkable.
It's totally wild. They mapped an enormous 4 megadalton molecular complex at a resolution of 3.74 angstroms.
The scale of that achievement is really hard to overstate. Just to give you an idea, in structural biology, a dalton is basically the mass of a single hydrogen atom.
So it's tiny.
Incredibly tiny. So a 4 megadalton protein complex is basically a towering molecular skyscraper. And measuring it at 3.74 angstroms means we aren't just looking at the rough shape of the building anymore.
Right, we're not just seeing the fuzzy skyline.
No, we are looking at the specific placement of the individual chemical bricks holding the walls together.
Which, it immediately begs the question, how do you even pull a molecular skyscraper out of a fragile egg cell without it just totally crumbling?
That was the huge hurdle. If you have a structure taking up 10% of the cytoplasm, you can't just throw the cell into a chemical blender.
Right, because you destroy the very thing you're trying to study.
Precisely. So the research team had to invent this remarkably gentle, low-dilution enrichment process.
Like a super delicate extraction.
Yes. They essentially coaxed these structures, which are known as cytoplasmic lattices, or CPLs, out of thousands of mouse oocytes using very minimal, highly specific detergent.
Oh, so they were dissolving just the cell membrane while leaving the delicate internal architecture totally intact.
Exactly.
So to give you a mental image, it's a bit like trying to extract a fully intact, wet spider web from a washing machine without tearing a single thread.
That is a perfect analogy. And once they actually isolated these delicate webs, they used single-particle cryo-electron microscopy, or cryo-EM.
Right. And for you listening, cryo-EM is so cool. It involves flash freezing the sample so rapidly, usually in liquid ethane, I think.
Yeah, liquid ethane is standard.
Right. So they freeze it so fast that water molecules don't even have time to form ice crystals, because ice crystals act like microscopic knives that would just shred the proteins.
Yeah, they would tear the lattice to pieces, but instead the water freezes into this glass-like state. It traps the proteins mid-action so researchers can take thousands of 2D pictures and stitch them into a highly detailed 3D map.
And that glass-like ice preservation was key to seeing the CPL clearly for the first time.
It was everything. Before this map, we knew that if the CPL failed to form, it led to a devastating condition called early embryonic arrest, or EEA.
Where the embryo just simply stops dividing right after fertilization.
Right, which results in permanent infertility, but nobody knew why. We didn't know what the lattice looked like, what it held, or how the pieces even connected to each other.
Okay, so let's start with the overall blueprint. When the software finally rendered that 3D image of a single repeating CPL unit, like the core building block that chains together to make the whole 10% web, what did it actually look like?
Picture a massive 36-nanometer spindle. It basically resembles an elongated diamond or maybe a microscopic football.
Okay, I'm picturing a football.
And the sheer complexity of this single repeating unit is staggering. It is constructed from 63 individual polypeptide chains, which are drawn from 16 distinct types of proteins.
63 chains just for one football unit.
Exactly. And when you look closely at its geometry, it exhibits what we call pseudo-C2 symmetry.
Oh, wait, I want to break down that term pseudo-C2 symmetry for
Okay.
listener, because if I'm visualizing this correctly, C2 symmetry means if you rotate it 180 degrees, it looks exactly the same, right? Like a playing card.
That's right. A perfect mirror image on rotation.
But the word pseudo means it's a trick. It's mostly a perfect mirror image, but there's like a slight lopsided wobble or structural difference tucked near the bottom.
Yes, and that lopsided wobble is absolutely no accident. The top half and the bottom half are fundamentally similar, but those slight asymmetrical deviations at the base are totally crucial.
Why? What does the wobble do?
They ensure that when these individual spindle units connect to each other to form the web, they only snap together in one specific direction.
Oh, so it's like a directional lock.
Exactly. It prevents the structures from accidentally bonding backward and creating this chaotic, useless traffic jam inside the cell.
That is so elegant.
It is. And to make sense of this 63-chain behemoth, the researchers categorized the spindle into 3 working zones. You have the external framework, the connecting linkers, and the central core.
Wait, so if I'm picturing this to keep the mental image really clear, is it kind of like a highly secure microscopic warehouse?
A warehouse. Let's hear it.
Well, like the external framework serves as the structural walls and roof, right?
Mm-hmm.
And then the linkers act like specialized bridges or highways, snapping thousands of these individual warehouses together into a massive grid.
Right.
And then that central core is the actual vault locked inside the walls where the precious cargo is hidden.
That warehouse analogy actually captures the functional reality perfectly. The external framework gives the structure its immense physical durability, which prevents it from being crushed by the chaotic cellular forces.
Makes sense. Got to protect the cargo.
And the linkers allow for polymerization, which is just the chaining together of units to form that massive web, taking up 10% of the egg.
And the vault.
The inner vault holds the biological ignition keys for embryogenesis.
Okay, let's do a quick mental recap for you listening. We are exploring the cytoplasmic lattice inside the unfertilized egg. So bullet 1, the shape is a massive 36 nanometer spindle.
Or football.
Right, a microscopic football. Bullet 2, it's built like a warehouse with external walls, connecting bridges, and an inner cargo vault.
And bullet 3, it has pseudo-C2 symmetry, meaning it's slightly lopsided to force a one-way connection.
Perfect. So keep that spindle warehouse shape in your mind's eye, because now we really have to look at the actual molecular bricks used to build those walls.
Because building a wall capable of surviving the chaotic environment of a living cell requires some serious structural engineering.
Oh, absolutely.
The researchers found that the long vertical edges of our warehouse, the main scaffolding, they are formed by a protein called PAD6.
And PAD6 doesn't just sit there all alone, right? The paper talks about decamers.
Correct. A single PAD6 protein just isn't strong enough on its own. So 10 PAD6 molecules fuse together to form a heavy-duty decamer complex.
10 of them? Wow.
Yeah, and 2 of these massive decamers arrange themselves into a short, right-handed helix just to build one single long edge of the framework.
So they twist together.
Exactly. This helical twist gives the wall incredible tensile strength, spanning about 22 nanometers in length. It is literally the unbreakable backbone of the entire system.
Okay, so the vault has its sturdy vertical walls, but a warehouse needs a solid roof and a floor to be fully enclosed.
Right, you can't leave the top open.
And the short edges, the top and bottom caps of the spindle, are built from a completely different set of materials called the subcortical maternal complex, or SCMC. Now, from what I understand, the SCMC is practically famous in reproductive biology.
Oh, it's a huge deal. It's the superstar complex of maternal inheritance. The SCMC sits right on the top and the bottom of those PAD6 walls, locking the vertical scaffolding together.
So it's the cap on the bottle.
Yes. Now, the SCMC is actually a team of 5 distinct proteins. You've got mater, TLE6, filia, ZBD3, and flopped.
That is a lot of acronyms to remember.
We definitely don't need to get bogged down in the alphabet soup, but you can think of them as specialized tumblers in a really complex molecular lock. And the most fascinating tumbler in this lock is flopped.
Yes. Okay, as I was reading through the source material, the behavior of flopped totally stopped me in my tracks.
So weird, right?
It's completely bizarre. So inside the SCNC cap, there are 2 copies of the flopped protein sitting side by side. You would naturally assume they just mirror each other perfectly, like identical twins.
But they don't.
No. Flopped is a shapeshifter. One flopped molecule uses its tail end, which is called the C-terminal extension, to fold into a really rigid, straight alpha helix.
Like a straight pole.
Exactly. But the second flubbed molecule sitting right next to it takes that exact same tail and folds it into a totally disorganized, flexible, squiggly loop.
Just floppy rope.
Right. And I'm just sitting there wondering why. Why would nature build the roof of this warehouse with a straight pole on the left and a floppy piece of rope on the right?
Because it is a masterclass in molecular docking. That physical asymmetry you just described, the pole versus the rope, is what creates two completely unique connection points on the outside of the warehouse.
Oh, I see.
And having unique connection points is absolutely essential for the linkers. Those are the bridges that connect one spindle to the next. The bridges are formed by a different protein called NLRP4F.
So because flub shifts its shape, it basically forces the linker bridge to adapt.
Exactly. Because flub shifts its shape, it forces the NLRP4F bridge to use completely different molecular hands to grab onto the warehouse.
Wait, I want to make sure we visualize this connection accurately. Let's avoid tech analogies like USB ports, because this feels much more highly mechanical.
It really is.
So is it kind of like a single Lego brick where the top has a standard rigid circular peg, but the bottom has like a flexible magnetic tether?
I like that.
And it forces the next piece to connect in a very specific alternating way.
That Lego analogy hits the nail on the head. The NLRP4F linker protein has distinct grabbing domains. On one end, you have what we call the PYD and NECHT domains.
And what do those look like functionally?
They act like a rigid claw that perfectly clasps onto the straight pole side of the cap. They lock in firmly with the philia and matter proteins.
Okay, rigid claw to straight pole. Got it.
But on the other end of the linker is the LRR domain. And that acts more like a flexible hand that reaches out and grasps the floppy rope side, anchoring into the ZBED3 protein.
Wow. And because it attaches using different hands on different sides, the connection is orthogonal.
Meaning it's perpendicular.
Right. The linker doesn't just stick two spindles together end to end like a long train. It forces them to connect at angles.
Yes.
And that geometry is what allows individual units to branch out and weave into massive, continuous 3D filaments throughout the cell, instead of just clumping up into a useless ball in the corner.
Exactly. Without that specific orthogonal twist, which is driven entirely by the shape-shifting flow ped, you don't get a web, you just get cellular debris. The entire 3D architecture is dictated by these tiny structural variations.
Okay, let's do another mental checkpoint for the listener, because that was a lot of detailed engineering. We've built the walls and the bridges.
Let's hear the summary.
Bullet 1, the long vertical walls are made of tightly coiled Patty-6 helixes.
Yep, the backbone.
Bullet 2, the roof and floor are the SCMC complex driven by our totally wild shape-shifting flow-ped-up protein.
The pole and the rope.
And bullet 3, the bridges connecting the warehouses are the NLRP4F linkers, which use different chemical hands to grab the straight and flexible parts of the roof.
Perfectly summarized.
So we've constructed the vault and chained millions of them together into this 10% web. But what exactly is the mother packing inside this thing?
Right. Well, now we step out of the scaffolding and into the central cavity of the core. And this is where the paper really redefines our understanding of the egg cell.
Oh, totally.
Because this vault is heavily fortified for a reason. It contains a highly volatile payload.
Here's where it gets really interesting. The first piece of cargo the researchers mapped inside the core is a protein called UHRF1.
Now, UHRF1 is an epigenetic manager.
What does that mean in practice for the cell?
After fertilization, when the DNA starts replicating, UHRF1's job is to read the genetic code and ensure that chemical tags, specifically methylation marks, are copied perfectly onto the new DNA strands.
Oh, so it decides which genes get turned on and which get silenced in the new embryo.
Exactly. It manages the operating system.
Okay, wait. I'm stuck on something here. I really have to challenge this logic.
Okay, go for it.
You're saying the mother cell goes through all the massive metabolic trouble of building this incredibly complex epigenetic manager, UHRF1, only to literally handcuff it inside a warehouse out in the cytoplasm.
That is what the map shows.
But that seems like a massive waste of cellular energy. Why build it at all if you're just going to lock it away? Why not just have the fertilized egg manufacture it later when the sperm actually arrives?
Because of the brutally tight timeline of early development, Synthesizing a complex protein from scratch takes hours.
Oh, and the embryo doesn't have hours.
Not at all. It needs to start managing its DNA the absolute second fertilization occurs. But you also can't just let an active UHRF1 manager wander freely around an unfertilized egg.
Why not?
Because if it slips into the nucleus too early, it will start prematurely methylating the egg's pristine DNA. It would completely ruin the genetic instructions before the sperm even gets there.
So it's not a waste of energy, it's a pre-deployment strategy. They have the manager dressed and ready for work, but they lock him in the vault for the cell's own safety.
Precisely.
And the paper details exactly how it's locked up, right? It's held in what they call an auto-inhibited conformation.
Yes. Inside the cavity, UHRF1 is completely surrounded by the PADI6 wall and two other proteins, UBE2D and NLRP14.
And they physically trap it.
They physically press against UHRF1, bending it so that its active site, the part that normally grabs DNA, is folded inward and blocked. It's a literal molecular straitjacket.
That is incredible. It's physically pinned down. Okay, let's look at the second piece of cargo. Because inside the vault, researchers also found 3 sets of alpha and beta tubulin heterodimers.
Now, tubulin is the basic building block of a cell's skeleton.
Right. When a cell divides, it snaps thousands of these tubulins together to build long cables called microtubules, which act like ropes to basically pull the chromosomes apart.
Exactly. But the researchers made a stunning discovery about how these specific tubulins are stored inside the CPL.
Okay, walk us through it.
Tubulin molecules require fuel to assemble into those long cables. They run on a molecule called GTP. Normally, as a tubulin brick is added to the microtubule cable, it burns through its GTP fuel at what we call the exchangeable site, converting it into GDP.
So the burning of the fuel is what locks the structure in place.
Right. But inside the CPL vault, the researchers found these higher resolution density maps proving that the tubulins are stored bound to GTP at both their exchangeable and non-exchangeable sites.
Wait. To put that in perspective for the listener, normally tubulin has to actively burn fuel to snap together. But here the fuel is still completely attached and unburned.
Exactly. It is the biological equivalent of storing a highly compressed, loaded spring. Or think of it like a rocket with the fuel lines already primed and the ignition switch pre-engaged.
So the moment the sperm arrives and the calcium wave triggers the vault to dissolve, these tubulins don't need to waste time hunting around the cytoplasm for GTP fuel.
No, they don't.
They are instantly ready to explode into rapid microtubule assembly. The cell division can happen at lightning speed.
The efficiency is just a marvel of evolutionary engineering. The mother provides a massive reservoir of preloaded structural springs.
It's genius.
It really is. And yet there is a third, equally dangerous cargo sharing that exact same vault space.
The paused shredders.
Yes. The central core also sequesters what is known as the SCF complex, specifically the FBXW and SKP1 proteins.
And what do those do?
These proteins are the targeting mechanisms for the cell's E3 ubiquitin ligase machinery.
Okay, I remember this. Ubiquitin ligase is essentially the cellular garbage disposal, right?
Basically, yes.
It slaps the chemical tag ubiquitin onto old or defective proteins, which signals the cell to just shred them into amino acids.
Exactly right. And during early development, the embryo has to rapidly destroy the old maternal proteins to make way for the new proteins encoded by the newly merged DNA.
So you need heavy-duty demolition equipment.
Yes, but again, you absolutely cannot have a protein shredder running wildly in an unfertilized egg. It would literally digest the egg from the inside out.
So just like the DNA manager we talked about, the CPL holds these SCF demolition complexes in a poised but completely inactive state.
Right. They are fully assembled, plugged into the wall, but sterically blocked.
So the safety switch is physically held in the off position by the surrounding lattice proteins.
Exactly. And it requires profound structural stability to maintain all that kinetic tension.
Let's pause and summarize this cargo haul for you listening. Think about the sheer amount of kinetic potential held inside this one microscopic vault.
It's explosive.
Totally. So cargo, 1 handcuffed DNA managers, ready to write the epigenetic code, but forcibly restrained to protect the pristine egg.
Cargo, 2 preloaded tubulin springs, fully fueled with GTP, just ready to explode into a cellular skeleton.
And cargo, 3 fully assembled protein shredders with their fingers paused right over the trigger.
It's a lot to hold back.
It really is. If we think about the insane tension held in this web, what happens if the locks break?
Well, if we connect this to the bigger picture, that question transitions this entire discussion from a fascinating biology lesson into a massive clinical breakthrough.
Really? How so?
Because when this highly engineered warehouse crumbles, the consequences for human reproduction are devastating.
Oh, right. Because these exact proteins like PAD6, the SCMC caps, the NLRP4F linkers,
Yeah.
they aren't just found in mice. Humans rely on this exact same architecture.
We absolutely do. And over the years, clinicians have sequenced the DNA of women experiencing severe, unexplained infertility, as well as families whose offspring suffer from MLIDs.
And for our listeners, MLID stands for multi-locus imprinting disturbance.
Right.
That happens when those epigenetic tags we talked about earlier, the ones managed by UHRF1, get completely scrambled.
Yeah.
It leads to severe, often fatal developmental disorders in the embryo because the genes just don't know when to turn on or off.
Precisely the issue. So geneticists have identified over 300 pathogenic or likely pathogenic genetic variants in these specific CPL proteins in humans.
300 variants.
Yeah. And for years, a doctor might find a missense mutation in a patient's PAA6 gene.
And a missense mutation is just a single typo in the DNA, right?
Yeah.
It swaps out one amino acid for another in the final protein.
Exactly. Just one tiny typo. The doctor knew it caused infertility, but they had absolutely no idea how one tiny typo could destroy a massive 4 megadalton complex.
But now, armed with this 3.74 angstrom 3D map, the researchers could literally run those human mutations through a highly advanced algorithm to see what happens.
Yes. They used a structure-based stability prediction software called MCSM.
MCSM, how does that work?
It's a graph-based algorithm that calculates the thermodynamic change in free energy when you swap out a single amino acid in a 3D space.
So it's basically a virtual crash test simulator.
Pretty much. It basically asks, if I replace this specific tiny brick with a slightly larger brick, does the whole wall fall down?
And the predictive power of that algorithm yielded staggering results, didn't it?
It really did. The software found that 85% of those known human missense variants act like a microscopic sledgehammer. They are predicted to physically destabilize the CPL structure.
So like a single amino acid change might sit exactly at the delicate interface where our shape-shifting flows protein connects to the linker bridge.
Exactly. And if that one amino acid is too bulky, the Lego brick just doesn't click. The connection is severed.
The warehouses fail to chain together, and the entire web just dissolves.
Which means the vault doors blow open prematurely.
The UHRF1 manager gets loose and starts scrambling the DNA tags.
The preloaded tubulin springs misfire into chaotic tangles.
The protein shredders go rogue and start digesting the cell.
Yeah.
And the result is early embryonic arrest.
It's catastrophic. But knowing this fundamentally changes genetic counseling.
Right, because this 3.74 angstrom map isn't just a cool 3D model for a textbook anymore.
No, it is a direct diagnostic template. A clinician can take a patient's genetic sequence, map the mutation directly onto this 3D structure, and say, look, your variant sits right on the ZBED3 docking site. Your lattices physically can't polymerize.
It provides a clear, mechanistic answer for patients who have been suffering from unexplained infertility for years.
It does, and it even opens the door for targeted therapeutics in the future.
Like if we know exactly which molecular joint is loose, maybe down the line we can design a drug like a molecular superglue to stabilize that specific interaction and save the lattice.
That is the ultimate hope of this kind of structural biology.
So what does this all mean for us? We started this deep dive thinking about the unfertilized egg as this quiet, passive factory floor, just waiting in the dark for a sperm spark to turn it on. But the reality is far more dynamic.
The massive paradigm shift here is how we define the cytoplasmic lattice. It is not just a passive box or a dusty storage unit. It is what they call a specialized proteostasis organelle.
An active ecosystem.
Yes. It is heavily regulated, constantly expending energy to inhibit, separate, and preserve the most volatile raw materials of life.
Which brings me to a thought I just can't shake. You know, we always define life by its kinetic energy, by movement, growth, cell division, and chaos.
Sure, that's the standard view.
But looking at this 4 megadalton volt, the very beginning of life requires an astonishing amount of perfectly engineered stillness.
That is a beautiful way to put it.
The egg has to hold all of these explosive, hyper-energetic molecules in a delicate suspended web. It is literally holding its breath, balancing on a razor's edge, just waiting for the exact millisecond of fertilization.
It's a biological contradiction. The potential for explosive, rapid growth relies entirely on the flawless perfection of that paused state.
Right, because if the stillness fails, the movement never begins.
So, next time you think about how life begins, I want you to consider this microscopic molecular waiting game. A skyscraper of suspended tension floating invisibly inside a single cell. What other cellular warehouses might be hiding in plain sight, just holding their breath? What stands out to you about the CPL's architecture? Think about that tension. Keep exploring the incredible machinery of biology, and as always, stay insanely curious.